today's lecture is on cells. And we're going to talk about cells at a lot of different levels. We're going to talk about the parts of the cell, which we touched on in lab last week. We're going to talk about genes. We're going to talk about cell division and how a gene gets turned into a protein and everything like that. And so when we're finished here today, you'll have a basis on how the cellular level works. And then next lecture, we'll take those cells, put the cells together in order to get tissues, and then we'll go above that. For the rest of the semester, we'll be talking about organ systems. So cell theory says that all organisms, all living organisms, are made up of cells. Whether that's a single cell organism or a multi-cell organism like us, they are made up of cells and things made by cells. So there is nothing in our bodies that is not related to cells. And a cell is the smallest part of our body that you would consider as alive. If you take a cell and you take part of that cell out, that part is not going to be alive. It cannot live on its own. A cell, in the right circumstances, can live on its own. We don't normally think of our bodies as being single cells that can live on their own, but that's what I do in lab all the time. I grow cells, and they live independently in a petri dish by themselves. Each organism structure in what that organism is capable of doing is going to be dependent on the cells in that organism. There are a lot of different types of cells. They each have a specialized function working together that's going to make your tissue, your organ, and your organ system. And all cells come from cells that are already existing. They're going to divide into two cells. The original one will eventually die, but then there's a newer one. We cannot make cells from scratch. And if you look at the cells from one species, compare it to another, compared to another, compared to another, they're going to be almost identical. If I have human cells under a microscope, you won't be able to tell the difference between that human cell and a mouse cell, or a rat cell, or a monkey cell, or an insect cell, or anything like that. When you get down to a cell, more or less a cell is a cell, and all of the chemical parts that we talked about last week are the same. The lipids, the proteins, the nucleic acids, they're all the same. So we're going to talk about a bunch of different types of cells, and we can more or less break these types of cells into three groups based on their shape. So here we have discoid cells. And so these are like the squamous cells that we talked about in lab last week. So they're flat. Here, if you kind of look at them from the top, they're kind of, again, flat. The red blood cells, we said, are kind of like a donut where the hole doesn't go all the way through. They're flat also. In all three cases, they sort of look like a disc. A fusiform cell is spindle-shaped, like this down here. We also have fibrous cells that look more like that. I honestly don't know why they included those four up there. I wouldn't try to label them as any of those. I think these three are definitely obviously discoid. This one is fusiform. That one is fibrous. Anything that you see as an example is going to be a prototypical, straightforward example like this. So a human cell is usually between 10 and 15 micrometers, micrometers. 1 micrometer is 1 millionth of a meter. You can picture what a millimeter looks like, right? If you can imagine a ruler with centimeters, those little markings between the centimeters are millimeters. And so 1 micron is 1,000th of a millimeter. So imagine that millimeter, break it up into 1,000 parts. That's 1 micrometer. And so a cell is about 1/100th of that millimeter. If If you took 100 cells, laid them side by side, it would only be about 1 millimeter. Egg cells, though, are very large. They're about 100 microns. And there are some nerve cells in our body that are over a meter long. And so while most cells are in this range here, we do have some that are much bigger. The reason that we don't have many that are so big is the fact that cells are in a three-dimensional world. And so as they get bigger, volume of a sphere is based on the cube of the radius. But the surface area is based on the square of the radius. So as you get bigger and bigger and bigger, the contents of that cell get bigger faster than the surface area would hold that in. So all of that water in that cell is heavy. So if it gets too big, the cell membrane is not going to be able to hold that in. So there are some basic components of a cell that we need to talk about. A lot of them we talked about in lab last week. One thing we mentioned, but we didn't specifically talk about, is the cell membrane, also called the plasma membrane. It's going to be the outside border of our cell. If it's going to hold everything in, it's also going to be a good landmark under the microscope to find the boundary between one cell and another. Usually under the microscope, if you have two cells next to each other, you won't be able to see the membranes of each. What you'll see is a dark line between two nuclei, and that dark line is going to be your cell membrane. The protein is mostly lipid that we'll see, although there are quite a few proteins in it also. Inside of that cell membrane is the cytoplasm. The cytoplasm as a whole is composed of the organelles, the cytoskeleton, which is sort of the bones of the cell. They give it the shape. We'll talk more about the cytoskeleton today. And then there is the cytosol. The cytosol is sort of the gelatinous liquid that everything in the cell floats around in. This is also called intracellular fluid, or ICF. Intracellular fluid is just because it's the fluid that is inside the cell. This is going to be opposite of extracellular fluid, which is liquid outside of a cell. The plasma membrane, we said, is the border of each cell. And if you have a powerful enough microscope, which we don't here, what you'll see is there's going to be two dark bands with a light space in between them. Like we said, the cell membrane makes the boundary between cells, and it also determines the interactions that one cell is going to have with its environment. It's going to talk to other cells around it. It's going to determine what comes into a cell, what goes out of a cell. This is a diagram showing this plasma membrane. And so we talked about these two dark bands. And what you're actually seeing there are these balls on the top and bottom of the membrane. And so the majority of this membrane is made up of these balls with two tails coming off of them. Those are phospholipids. We talked about phospholipids last week. It's a polar molecule where the one side is hydrophilic, meaning it likes water, and these tails are hydrophobic, meaning they don't like water. And you get this double layer where, so because the tails both don't like water, they turn towards each other, and the heads that do like water will turn towards the extracellular fluid and the intracellular fluid. And so the dark lines are the heads, and then the light space in this picture is where the tails are. 98% of that membrane are lipids, most of which are phospholipids. So 75% of the membrane is phospholipids. We said that they have one end that likes water, one end that doesn't. The term that we use to describe that property is amphipathic. Like amphibian is land and water, ampha is both, and then that should be a T there. Amphipathic. And so the hydrophilic phosphate heads, the balls, face the water. The hydrophobic tails are in the center of the membrane. These float around on each other though. If we looked at this picture, everything would just be randomly moving around. Those phospholipids move around fluidly. There's also cholesterol in our cell membrane. And so that is these blue molecules here. And cholesterol is about 20% of the lipids. The purpose of the cholesterol in our plasma membrane is to hold the plasma membrane still. We said the phospholipids can drift around. If they drift around too much, they don't really have any shape to them. Cholesterol is kind of a rock that anchors itself to the phospholipids around it and holds them in place. So that way our cell membrane at least has a little bit of stiffness to it. There's also some glycolipids. So we said a glycolipid is a lipid that has a sugar attached to it. And so of the lipids in our membrane, about 5% are glycolipids. Most of these are phospholipids that have a carbohydrate stuck to the extracellular face. So that's the head that faces outside of the cell. And the main purpose of this is to make what we call the glycocalyx. Around the outside of a cell, there's a carbohydrate coating, and its primary purpose is communication with the other cells around it. So if another cell bumps into it, it can see what sugars it's showing, and that will tell the second cell what type of cell that it bumped into. There are a fair number of proteins in the membrane though, and the proteins are going to really be functional. The lipids are structural, the proteins are functional. Even though if you just counted all the molecules in the membrane, only about 2% of them would be proteins. If you take them by weight, proteins make up 50% of the membrane. Lipids are small and light, proteins are large and heavy. There's a number of different types of proteins that we're going to talk about, and we can break them down into one or two subtypes based on where they are in relation to the membrane. So an integral protein goes through the membrane. So this is an integral protein. These are integral proteins. They are going through the membrane. If you have a protein that goes all the way through, like we have here, that's called a transmembrane protein. Each part of these proteins are going to have different likenesses to water. And so the parts of the protein that are buried in with the tails of the plasma membrane are going to be hydrophobic. They're not going to like water. The parts that stick into the intracellular part and the extracellular part will be hydrophilic. They like water. So the protein will orient itself so that each part ends up around the type of molecules that it wants to be around. If you have a protein that does not penetrate into the membrane, it's going to be called a peripheral protein. It stays on the periphery of the membrane. And so that is down here. And so it is right up next to the plasma membrane, but it's not actually in the plasma membrane. And so usually these are held in place because they're attached to the cytoskeleton, which we still haven't talked about. But this is these proteins down here. It's forming kind of a skeleton under the plasma membrane, holding it in place. This peripheral protein is going to be stuck to that. So functionally, it's part of the plasma membrane, but it's not actually in it. We're going to talk about all these types of membranes more in depth. They each have their own bullet points. But you can, looking at this picture, you can really see the various roles that these proteins are going to play. So a receptor is a protein that's going to sit there in the membrane, and it has a little slot on the extracellular part that fits a specific molecule. And if that molecule comes around and it sticks in there, the cell then knows that it saw that signal. It's going to do something on the intracellular part down here to send a signal that this messenger, chemical messenger, has been found. An enzyme is going to be a protein that does something. It has a functional purpose. It changes another molecule. I don't really like the definition that they have here. It's a little too strict. The next slide will have a little bit broader definition. A channel protein is going to be a protein that has a permanently open channel through it. And so molecules and atoms can go through that channel. It may be non-specific. It may allow anything that'll fit to go through, or it may be specific, allowing only certain molecules to get through. But it doesn't actually move. It doesn't pump anything. It doesn't open and close. It's just permanently open. We also have channels that can open and close, and that's a gated channel. Depending on what it wants to get in, it can open and close. If it sees something that it wants to let in, it can open. If there's already enough of it inside, it can close. Here's a protein that has a carbohydrate stuck to the outside of it. Again, that's going to be a marker telling other cells what type of a cell this is. And then there are cell adhesion molecules which reach out and touch a cell adhesion molecule of another cell. They grab onto each other, and that holds those cells together. So we said a receptor is going to bind a chemical signal. Second messenger systems weren't mentioned on the previous slide. There was no picture of them. A second messenger system is usually what happens once a receptor gets activated. And so that ligand comes in and binds to the outside of that receptor. The receptor gets turned on, and it activates this messenger system inside of the cell. That's going to cause a chain reaction of events that's eventually going to usually lead to a change in a gene expression. It's going to turn a gene on or off. So enzyme is going to catalyze a reaction, period. That reaction may be the digestion or breakdown of a molecule. It may be the production of a second messenger. But an enzyme is going to catalyze a reaction. Whether that's making something or breaking down, it's still an enzyme. So then we have our channel proteins. So it's going to allow molecules and atoms to get through the membrane. We said some are always open and that some are gated. The ones that are gated have a few different mechanisms that they can use to determine whether they need to be open or closed. A ligand-gated channel is going to respond to a molecule binding to it. So imagine a receptor where there's a spot on the outside for a molecule to bind, and when it binds, our channel opens up. So that is going to be a ligand-gated channel. A voltage-gated channel is going to detect changes in charges inside and outside of the cell. And it's going to want to maintain a specific charge. If that charge changes, it's going to open up in order to restore the charge. We're really going to see that when we get into neurons and nerve function. Then there are also mechanically gated channels that if you actually push on them, they're going to open up. So this a lot of times has to do with the amount of water in a cell. If there's too much water in a cell, it's going to put a lot of pressure pushing out on that cell membrane. And so these are going to detect that, say there's too much pressure, I don't want the plasma membrane to break, and so I'm going to open up and let some water escape. A carrier is similar to one of these channels, but this is more like a pump. A channel has just a hole that things can move through, and it can open and close in some cases. A carrier is going to grab something either on the inside or the outside, and forcefully move it to the other side. This is going to require energy to make that happen. A channel does not require energy necessarily, but a carrier or a pump is going to require energy, and that's going to come in the form of ATP. These are our cell identity markers that we talked about. These are usually glycoproteins, a protein with a carbohydrate attached to it. And then we talked about our cell adhesion molecules that hold cells together. There are a few different ways that cells can be held together. The first one is going to be called a tight junction. In a tight junction, the cell membranes of two cells are basically fused together in one very thick double layer plasma membrane. Usually, in things like our cells or our liver, if we have two cells of the same type, you're usually going to have a tight junction that are going to hold those two cells together. And so this is a tight junction. So what we have here are two cell membranes. So there's one cell that's over here, another cell that's over here, and then there's two membranes that come together next to each other. A tight junction is basically going to attach those together. It's like a rivet holding them together. The plasma membranes actually touch where those connections are. So this is not going to allow anything, including water, to sneak between those cells. So if you have something that's going to be used for filtering, like your kidneys or your liver, you're going to have a lot of these. That way, water with toxins can't sneak through the cells. In order to get to the other side of the cells, they're going to have to pass, the water is going to have to pass through the cell. And the cell at that point can decide what it wants to let through and not. The next type of a connection is called a desmosome. A desmosome is basically those cell adhesion proteins. You have proteins on the surface of two different cells. They're going to reach out and grab each other and hold on. These are going to be reversible. In certain conditions, the cell can do it. Other cells around it can make this release. If I grow cells in the lab, they use this mechanism to attach to the petri dish. All I have to do is add an enzyme. I can either add an enzyme to digest the protein, that way it can't hold on anymore, or I can just add something that takes all the calcium out of the water. These proteins need calcium. So if a cell gets rid of the calcium around it, it's going to let go. So that is the desmosome. It's just two proteins holding on to each other for as long as they want to. They can let go if they want. The third type is called a gap junction. This is similar to a tight junction, but the protein holding them together is hollow. And so there is actually a pathway from one cell over here over to the other one. So this is going to allow one cell to communicate with the one next to it. Molecules can move through those pores. Questions about anything so far? Yes?
Just out of curiosity, those gap junctions, do those create ATP? Or not at all? It is?
No, they're not active, and so they're like a channel, and so there's just a hole there. They just sit there, but because they're kind of circular, like a tube shape, so they have a hole going right down the middle.
You said tight junctions only let water through?
A tight junction cannot let anything, including water, through. So a tight junction, nothing can go through that spot between the cells. So if you have a layer of cells and water wants to get to the other side, it's going to have to go through the cell. The plasma membrane itself is more permeable than these junctions holding the cells together. So we talked about microvilli, cilia, and flagella in the lab. Are there any questions about those? It's the same exact slides that we went over in the lab. But now we're going to talk more about getting things from one side to another of the plasma membrane. So the plasma membrane is selectively permeable, meaning that there are some atoms and molecules which can just pass right across it. Most things, though, cannot. If you're going to get something across that plasma membrane, you can either allow it to happen passively, meaning it's going to happen, you don't need to do anything, it doesn't require any energy, or you can do it actively, meaning you're going to make something happen that doesn't want to happen, and so you're going to have to use ATP energy to make it happen. And so passive mechanisms are just based on the fact that molecules move around randomly. Molecules in the gas form or liquid form, they just float around. They're going to float around and they're going to go where they want to go. That's going to happen regardless of what you do. And so them moving on their own has enough energy to make these passive mechanisms happen. This includes filtration, diffusion, and osmosis. We'll talk about each one individually. And then there are the active mechanisms which require ATP. And this is going to be the active transport, which is membrane proteins, and then a completely separate process called vesicular transport. If you have a carrier-mediated mechanism, that is going to be a membrane protein that uses ATP to transport substances across a membrane. So this is a subtype of active transport. So this is filtration. Filtration uses pressure to push something through a filter. And the example we're going to use is water going through the walls of our capillaries. So this is a very blown up picture of a capillary. And so we have blood cells and plasma inside of our capillary, and then we have other cells forming the wall of the capillary. But you can see they don't fit together all that well. There are very small gaps here. And so if you have pressure inside of that capillary, it's going to force water out. Yes, water will find its way through a hole on its own, but if you pressurize the inside, it's going to force that water out. And so big things like cells are going to stay inside the capillary, then when they can make their way back to the main blood supply, get reoxygenated, and make another go-around. Smaller things, though, like water and nutrients, are going to get through that capillary and then move on into the cells around that capillary. So a capillary is a very small blood vessel that goes right into a tissue. And so the blood is going to leave a big blood vessel, go into the capillaries, and then the nutrients in the water will leak out into the tissue. And then it will have to go through the plasma membrane into an actual cell. So this allows that to happen. It also allows waste to be removed from the plasma. So this happens in the kidneys. And so imagine you have this capillary in your kidney. Instead of wanting nutrients to get out into cells, you're going to force the water containing the toxins out of the blood. Once they're out of the blood, then the kidney can process them, and the good things get left behind and just keep on their merry way. So diffusion is just the movement of chemicals and molecules from a place where there's a lot of them to a place that there's a small amount of them. If we open a bottle of perfume in that side of the room over here, there's going to be a very high concentration of perfume over here and a very low concentration over there. But you know that if I leave that open, eventually it's going to make its way over there. That's diffusion. It's just where there's a lot of something, what there is there wants to move to where it isn't. think of molecules as being antisocial. They want to get away from each other. They want to find peace and quiet. And this is based off that fact that those molecules are always moving. Those perfume molecules come up into the air and they fly around randomly. If they fly around randomly, eventually they're going to make their way over to that side of the room. We talk about things moving down or up the concentration gradient. So a gradient is a smooth transition from high concentration to low concentration, or high temperature to low temperature. It's not usually stepwise, it's usually a smooth transition from high to low. And just like a ball rolling down a hill, things are going to go from high concentration to low concentration. If you want something to move down its concentration gradient, that is the direction that it wants to go. And so all you have to do is not get in its way. And so you don't need a membrane to do this. You don't need a protein to do it. You don't need anything. If you do have a membrane, though, you're going to block this from happening unless whatever is diffusing can get through that membrane. So a plasma membrane gets in the way. It's going to allow very small things like nutrients to diffuse across, because we want those to get into the cell. But it's going to prevent other things, like large molecules, from getting through. Similar to this is osmosis. Osmosis is, think of it as a specific circumstance of diffusion. So osmosis is the movement of water through a selectively permeable membrane. So a selectively permeable membrane means a membrane that allows certain things through, but not other things. And so in the case of osmosis, water can pass through this membrane. Other things like salt and sugar cannot. So in this case, really the only thing that's going to move across is the water. And so the water is going to move from one side to the other. And you remember that water always wants to move towards the higher solute concentration. If we have a membrane and there's a solution on each side of that membrane, the water is going to move across that membrane to get to the higher concentration. Essentially what's happening is when you have a lot of solute particles dissolved on one side, they're going to draw the water over to the other side. And so this is what's going on here. So we have a beaker, and right down the middle of our beaker is a membrane. We have two solutions, one on side A, one on side B. Side A has a whole bunch of solute, let's say it's sugar, dissolved in it. Side B does not have that sugar in it. And so first of all, which side has the higher concentration of solute, A or B? A. And so water is going to move towards the high concentration. And so the water over here is going to come through the pores over to this side. And so what you end up with is actually more water over here than over here. The water moved from that side, the right to the left, and so the level on the left went up, the level on the right went down. We're not going to talk about this osmotic pressure and hydrostatic pressure, so you can cross that part off. What you need to know is that water is going to move across the membrane towards the high concentration of solute. And this is really important if you're going to give a patient IV fluid. Imagine you put an IV into a patient and you pump really high concentration of saline into their body. Plasma membranes are semi-permeable membranes. They're selectively permeable membranes that water can pass across. And so in that case, you're going to have a very high concentration of salt in the plasma, a very low concentration in the cells, and so the water is going to come out of the cells into that plasma. So we said I just said that water can go across a plasma membrane, but it's kind of slow. Our cells need to be able to respond quickly though. And so they have proteins, these are channel proteins, called aquaporins. Aqua, water, porins, or pores, or holes. They're just open channels that let water move through very quickly. And so when we talked about that IV fluid, the comparison of a solution outside of a cell to the concentration of a solution inside of a cell is called tonicity, or tonicity, which, however, is easier for you to pronounce it. This is the effect of a solution outside of a cell on the amount of water inside of a cell. And so a hypotonic solution, hypo meaning low, like hypothermia, is a solution that has a lower concentration than the solution inside of the cell. And so a cell, I think it's down at the bottom of the slide, but I'll put it up here. A cell has a concentration of 0.9% sodium chloride. So a hypotonic solution is going to be a solution that has less salt than that. So if I have a solution that's 0.5% saline, that is going to be a hypotonic solution. So you have a high concentration in the cell, low concentration outside the cell. Which direction is the water going to flow? It's going to go towards the high concentration. Where is the high concentration? It's in the cell. And so water outside of the cell is going to move into the cell, which is going to make it blow up like a balloon. Our cells are designed to handle that for a little while. If they stay like that, they're going to cause damage. If it blows up too much, it'll actually burst, and then obviously there's no coming back from that. The opposite is a hypertonic solution. Hyper meaning high. So in this case, let's say we have a 1.2% solution outside of the cell. In this case, which direction is the water going to flow? Outside. It's going to come from inside the cell to the outside. So the cell is going to shrivel up. If it shrivels up too much, it's going to die. If you have a solution that is just the right concentration, that's called isotonic solution. Iso means same. So in this situation, water is going to move back and forth across that membrane. You can never stop that from happening completely. It's going to go both directions at the same rate or the same speed. And so the amount on either side is never going to change. And so these are our red blood cells in three types of solution. So in an isotonic solution, this is what a red blood cell should look like. If you put it into a hypotonic solution, the inside of the cell is then the high concentration. So water outside the cell is going to flow in, making it blow up and swell up. If you put it into a hypertonic solution, then the high concentration is outside. And so the water that started inside is going to come out, and our cell is going to shrivel up. So crenated is the technical term for shriveled up. Shriveled up is fine by me. You don't need the technical term. Okay, so that was passive mechanisms. Things that are just going to happen on their own and don't require energy. Next, we're going to talk about the carrier-mediated transport. So these are proteins buried in that plasma membrane that are going to move solutes into or out of a cell, or some organelles actually have their own membrane proteins. So they're going to pump things in or out of an organelle. It works exactly the same though. It's just a plasma membrane with a protein buried into it. So most of these proteins are going to be specific, meaning that they only transport one or two different things. They're not going to be generic. They're made to transport one or two specific things. The solute is going to bind to the receptor, either on the inside, if it's going to get pumped out, or the outside, if it's going to get pumped in, and that's going to cause the protein to do something to move it into the other side. But the solute does not get changed. This protein is not going to do anything to the solute other than move it. It's not going to break it down. It's not going to make it bigger. All it's going to do is move it. And you can imagine if these membrane proteins have to actively do this, at some point, if you have a whole bunch of solute, they're not going to be able to keep up. And so that is called saturation. If you have so much solute that needs to get to the other side of that membrane that the protein can't keep up, we say that it's saturated. And so how fast it's going to happen is going to increase as you have more and more solute until it maxes out. And that point where it maxes out is going to be called transport maximum. This is not a term you need to know, but you do need to know the idea of saturation.
Can you say it again?
Yeah. And so imagine you have a membrane, and then you have some proteins here. If you only have a couple, 2 or 3 solute particles that these need to pump across, they can keep up with it. But if you have a whole bunch of them, these are just going to pump and pump and pump as fast as they can, but they can't keep up. So we say they're saturated. They can't pump any faster. Just like a sponge is saturated when it can't hold any more water, these pumps are saturated when they're already going as fast as they possibly can. So my boat has a pump in it. If I forget to put the plug in the boat, or if I spring a leak, it's going to pump water out. As long as I'm leaking slower than my pump can pump, I'm okay. But if it starts leaking really bad and my pump can't keep up, my pump is going to be saturated. And so it's pumping as fast as it can, but the water's still coming in, and so I'm in trouble. That's basically what's going on here. And so these types of proteins come in 3 flavors. There's a uniport system and a symport system, which work both basically the same. It's just a uniport carrier only carries one solute. There is a calcium pump, which exists for the sole purpose of moving calcium. So uni being same or one. Uniport carrier is only for one solute. A symport system, sim meaning same, is going to move at least 2 solutes at the same time in the same direction. So it's going to pump 2 or 3 things in, it's going to pump 2 or 3 things out. But there has to be at least 2 things, and they have to be moving in the same direction. An example we're going to see is a sodium glucose transporter. Sodium has a very strong force that it wants to get into a cell. And so these symport carriers are going to use that and say, okay, sodium, I'll let you in, but when you come in, you have to bring glucose with you. And so that's going to get glucose in. So sodium and glucose will get transported at the same time in the same direction. The opposite of that is an antiport system. This is going to move, again, at least 2 things, but they're going to go in opposite directions. One thing is going to come in, another thing is going to come out. A sodium-potassium pump is going to pump sodium out of the cell while pumping potassium into the cell. One thing you're going to see over and over again throughout the semester is that inside of a cell has a lot of potassium. Outside of a cell has a lot of sodium. And so these processes can work through facilitated diffusion, meaning it's just going to happen, or they're going to be primary active transport or secondary active transport, which we will see. And so facilitated diffusion can work in here. Is it's just going to let solute move down its concentration gradient. Imagine sodium wanting to get to the other side of a membrane. It's going to bind to the outside of the protein, and then it's going to pump it to the other side. That's facilitated diffusion, and that is not going to need any energy to make it work. So the solute is going to attach to the outside. It's going to make it change shape. Remember, conformation is the shape of a protein. And so essentially you have something that's open like that. The solute is going to come into that V, and then it's going to change shape to release it to the inside. And then once it's released, it's going to go back to grab another one and just keep going back and forth, releasing things into the cell. Primary active transport is going to move a solute up its concentration gradient. So it's going to move it opposite to the direction that it wants to go. In order to make that happen, it's going to have to use ATP. If you want to make something do something that it doesn't want to do, you're going to have to use energy to make it happen. So that calcium uniport pump has to use ATP. It's going to export or expel calcium from inside the cell to outside of the cell. And the calcium is already more concentrated outside of the cell. So calcium wants to come in, but this pump is going to use ATP to pump what is inside out. The sodium-potassium pump is going to use ATP to pump sodium out, and in that process is going to allow potassium in. So this is that sodium-potassium pump. Let me get everything up here. So this is our protein. And in this picture, the orange atoms or ions are the sodium. The blue ones are potassium. So starting out, we have a bunch of orange sodiums outside of the cell. And we have a bunch of blue potassiums inside of the cell. The orange wants to go in, the blue wants to come out. Our protein, though, is going to use ATP, and when it does that, it's going to take 3 of the sodiums from inside, pump them out, and it's going to pump 2 potassiums in. So with 1 ATP, it's going to take 5 ions in total and pump them where they don't want to go. So it's going to keep a high concentration of potassium inside. It's going to keep a high concentration of sodium outside. Sodium can slowly leak through a plasma membrane. So you're pumping them out while they're leaking in. And so these pumps can never shut off. Our cells require very little sodium inside and a lot of sodium outside. And so these pumps are always running, and they actually use up half of the calories you burn in a day. If you just sit around and don't exercise, half the calories you burn that day will be used simply by these pumps, pumping sodium out of your cell, pumping potassium into your cell. Just to maintain that balance, you're going to burn a whole bunch of calories. So we'll talk about secondary transport in a minute. The reason we want to control these ions is because the more sodium you have in a cell, the more water you're going to have in a cell. And so to maintain a specific amount of water in your cell, the cell needs to control how much sodium is there. If this stops working and the sodium leaks in, your cell is going to expand and expand and expand, and eventually it's going to burst. It also maintains a membrane potential. Well, again, we'll talk about that more in depth in the neurological section. but it's a charge difference from one side of the membrane to the other, and these also make a lot of heat. Our body heat comes from muscles, yes, but these pumps also generate a lot of our body heat. So secondary transport is similar to primary active transport in the fact that it requires ATP energy, but it's going to use the ATP indirectly. And so it's going to require primary active transport to happen first. And the example we're going to use here is the sodium glucose transporter. And so the sodium glucose transporter is a symport system, so it's going to move sodium and glucose in the same direction. And in this case, it's going to be pumping them into the cell. And it says it depends on the primary transport performed by the sodium-potassium pump. Why does this require this to be working? So we're going to pump sodium from the outside in, and at the same time, we're going to pump glucose from the outside in. If the sodium-potassium transporter was not working, what would happen to the amount of sodium on both sides of the membrane eventually? It would leak in, and then it would be the same. So if there's the same amount of sodium on the inside and outside, sodium doesn't want to come in. This sodium glucose transporter is based on the fact that sodium wants to come in. And so the cell can say, okay, sodium, you can come in, but you better bring glucose with you. And so if this stopped working, sodium doesn't want to come in, and so you can't talk it to bringing in glucose with it. So it requires the use of ATP, but it doesn't use it itself. If the cell ran out of ATP, the sodium-potassium pump would stop working, and so eventually the sodium-glucose transporter would stop working.
So because of bringing in glucose, why would that process stop? Breaking into the pyruvic acid and all that stuff.
Why would the sodium-glucose transporter stop?
Why wouldn't it be able to break down the glucose?
It could. It just would run out of glucose. So it could break down the glucose that's in the cell, but as it breaks it down, it has to pump more in from our blood.
Oh, so it's pumping in.
Right, so there'd be plenty of glucose in our blood. It's just the cell has no way of getting it into it in order for it to break it down. So this is a cell. And in this case, we actually have the sodium glucose transporter on one surface. And down here, we have the sodium potassium pump. And so the sodium potassium pump constantly pumps the sodium out, which makes there a very low concentration of sodium inside the cell. And so the sodium up here really, really wants to get in. And so the sodium glucose transporter is going to let one sodium in if it brings a glucose with it. And so by having these on opposite sides, our kidney is going to prevent glucose from getting out of our urine, because the urine is going to be down here. Glucose is going to be pumped in up here, and then it's going to get burned up here before it can get down here to the urine. The next type of transport is going to be vesicular transport. So this is very, very different than the things that we've talked about before. This is going to use vesicles that we talked about in the lab last Wednesday. So remember, a vesicle is a sort of a membrane sac that comes off of the Golgi. It's just a sphere of plasma membrane that has water and whatever is dissolved in that water in the center of it. It's basically a water balloon. And so vesicular transport is going to be used to move large things. Pumps and proteins can move molecules. Vesicles can move large things. It can move organelles. It can move whole drops of water at a time. And so we can use it to bring things into a cell also. We'll see how that works. We can use it to bring in water from outside the cell. And we can also use it to release things. So the process of using a vesicle to bring something into the cell is called endocytosis. Endo is inside, or gaining something. The cell is gaining something with endocytosis. And that has three subtypes. Number one is phagocytosis. And so that is going to be a cell eating something big that's outside of it. This is primarily going to happen with your immune cells. If they're floating through your bloodstream and they come across, let's say, a bacterium, your cell is much bigger than the bacterium. And so it's going to fold its plasma membrane around that bacterium and just swallow it up. Penocytosis is cell drinking. And so it's going to have the same process. It's just like it's eating something, but what it's eating is just a bunch of water. And so there's going to be nutrients included in that water, because the water is not just water, it's extracellular fluid. It's things that have come out of the capillaries in your tissue. So there's nutrients and things in that water that the cell can then bring in. Some of these are going to be receptor mediated, meaning there are proteins on the cell membrane that act as receptors, and if the right molecule binds it, it's going to trigger this to happen. And that's going to make this what we call a clathrin-coated vesicle, which we'll see in a minute. The opposite of endocytosis is exocytosis. It is the same process in reverse, and we'll see what this looks like in pictures. That's going to release things from the cell, and all of this uses proteins that act as motors and require ATP. Vesicular transport requires a lot of energy, but it's worth it because it can move large things. So this is endocytosis, particularly receptor-mediated endocytosis. So we have a plasma membrane here, and then we have molecules that are going to bind to the receptor, and that's going to tell the cell to bring them in. And so when it binds, the cell is basically going to fold the membrane on itself until it actually touches here, and then it's going to pinch off. And once it pinches off, the plasma membrane is still continuous. There is no hole in the plasma membrane. We now have this vesicle inside of the cell that it can take in, and it can eat the things that it just brought in. And so this is called the clathrin-coated vesicle. I don't worry too much about the clathrin part. You need to know the vesicle part. And so it's going to form a vesicle to bring something into the cell. The opposite, we said, is exocytosis. It looks exactly the same. So here, this vesicle is coming from down in the cell. It's going to come up until it touches the plasma membrane, and then it's going to fuse with it. And when it fuses, now the plasma membrane was just up here. Now, that is the plasma membrane. So now what was in that vesicle is now open to the outside. And so it's going to use this to release things to the outside. This has two purposes. The cell can use this in order to release waste, things that it makes that it doesn't need, it wants to get rid of. It's just going to dump it outside of the cell. But this also needs to happen if endocytosis is going to continue to happen. A cell only has so much plasma membrane. If it keeps swallowing it up, the cell is just going to keep shrinking and shrinking and shrinking and shrinking. And so for every vesicle that comes in, another one has to come back in order to replenish the plasma membrane. So sometimes, even if a vesicle doesn't have anything that it needs to dump out, it's just going to come to the plasma membrane empty, because it has to keep replenishing that plasma membrane. Okay. Questions on anything yet?